Chuck Bronte. So one of the key motivation behind this study strikes a common chord with a lot of the talks we've seen today already, and that is trying to understand how Lake Michigan salmon and trout are adjusting to a changing forage base. As we've heard repeatedly today, the pelagic forage fish in Lake Michigan have been in decline since the 2000s. We've seen this for alewife, for bloater, and for rainbow smelt. And we've also seen more recent declines in native benthic forage fish with deepwater sculpin and slimy sculpin, as well as fluctuations in invasive round goby. All of these figures are from the USGS report from last year's meeting. So understanding how niche partitioning and potential for competition among the salmonine species during these shifts in forage abundance is going to be critical for fisheries management. And so our approach for this is a stable isotopic approach. We chose this approach for a number of reasons. First and foremost, stable isotopes of carbon and nitrogen offer a time-integrated assessment of diet. Gut contents remain a very important tool for assessing diet, but they do provide information over food consumed over the last several hours to days. By contrast, stable isotopes basically reflect the diet over the past several weeks to months, so it gives a longer time period of the diet that is reflected in those isotopic signatures. Stable isotopes can also help capture complex interactions and help to visualize energy flow through the entire community. And because this method has been around for several decades in aquatic ecology, there are refined tools now for assessing diet and comparing niches. So we were able to pursue this through the mass marking program, in large part because of the large field effort that goes on every year as a part of the tag recovery program. The biotechnicians handle over 20,000 salmonines every year from Lake Michigan, and that spans a broad geographical area lake-wide. So the sample collection can occur at no additional cost, and we can collect enough samples to really capture the variability in stable isotopic signatures of these animals. So this allows us to also investigate details such as regional differences, temporal differences, size class differences, and differences between stocked and wild fish. Now, I won't have time to get into these today, but there are some interesting details coming out of this. For example, we do see some regional differences in lake trout and brown trout signatures. Hopefully we can talk about that in more detail in the break if anybody is interested. In terms of how isotopes are interpreted, the carbon ratio does give us an indication of nearshore versus offshore samples. And the nitrogen isotope ratio gives us an indication of trophic level. In Lake Michigan, it also gives us an indication of offshore benthic items, which tend to be more enriched in nitrogen. And to help explain how we interpret these isotopes, I'm going to show you, this is a biplot showing the isotopes from the 2014 collections from the mass marking program. I do want to thank Ben Tershak and Harvey Bootsma for providing 2 data points here for amphipods and for small alewife to complete this figure. And so first, in terms of nearshore versus offshore, we do see an enriched carbon signature for amphipods and nearshore round gobies. And we can contrast that with some known offshore items that are more depleted in carbon that stems from zooplankton, which you see at the lower end of the figure, but also you see alewife and represented by the ALE symbol, and at the very top, this top circle here, it's not going to work, the DWS and SS are the sculpin species. So we do see that near versus offshore distinction. In terms of trophic level, we can pull out a classic food chain from this figure. We see zooplankton, followed by alewife and smelt, and then even higher than that are the salmon and trout species. And so we do see about a difference of about 3 units of nitrogen for each of these steps, as one would expect for this kind of increase in trophic level. In terms of the signature for offshore benthic being more enriched in nitrogen, I want to point out the difference here between alewife in this red circle and the sculpin species in this red circle. The sculpins have a more enriched nitrogen content than even the majority of the salmon and trout species, and I don't think anybody in this room would claim that sculpin are a higher trophic level than salmon and trout. So it makes the most sense that we do see this enrichment in nitrogen offshore, which has been shown by prior studies as well. So So this brings us back to our key motivation. How are Lake Michigan salmon and trout adjusting to the changing forage base? What I want to show now are two density plots. This is a density plot of the carbon signatures, and what we see here is that all 5 of these species share a very similar carbon-13 signature, but there is a fair amount of differentiation in nitrogen. Specifically, the lake trout have an enriched nitrogen by about 2 parts per mil compared to the other 4 species. Now, this may not sound like much, but if we go back to our lake-wide biplot, the difference between the lake trout and these Oncorhynchus species of about 2 parts per mil is about the same difference as between those Oncorhynchus species and the large alewife. So that is a substantial difference in nitrogen for lake trout, showing that they have a more unique niche. And in my mind, it suggests that lake trout have a greater reliance on offshore benthic prey, which has been touched upon by a number of talks today already. We can perform a diet mixing model based on the isotopic results to provide some insight on what the potential diet might look like, and this does substantiate this idea, where we do have an estimate here of about 40% of the lake trout diet being comprised of offshore or benthic prey items. By contrast, the 3 Oncorhynchus species, the Chinook salmon, the coho salmon, and rainbow trout, all have 80 to 85% of their diet coming from alewife and smelt based on our diet mixing model. We also see round goby contributions to all of the diets except for Chinook salmon. At first I was a little surprised to see this estimate for coho salmon, but I did speak with a colleague yesterday who has seen gobies and coho diets as well. So there is going to be a reliance on this resource for some species. And then one thing that doesn't come out of this table that I do want to point out is we were able to break down the alewife from between small and large alewife, and as it turns out, it looks like the lake trout, our mixing model is suggesting they're consuming larger alewife, where the other species are consuming smaller alewife. Again, this is suggested by the mixing model, and we do need to corroborate this with diet analysis. But these 2 figures here just show the proportion of the diet as estimated by these mixing models for small alewife on the left and large alewife on the right, with the consumers along the x-axis. And so that difference in between lake trout and the other 4 species becomes apparent. One caveat I want to point out with these diet mixing models is they're very sensitive to the isotopic signature of the prey, and it so happens that prey signatures vary substantially by depth and size. So just a classic example of this, if you look at that cyan, that really light blue, that's the round gobies, and we see a carbon signature spanning from about -16 to -25, which is consistent with this idea that round gobies are increasingly occupying offshore areas, and they're going to span that nearshore to offshore gradient. So just keep that in mind when thinking about these mixing model results. One thing we can do that's independent of estimating diet but still assesses niche overlap is this new method that uses probability to directly measure overlap. This is based on an ecology paper published by Swanson et al. Last year, and what it essentially does, this is a scatter plot of all the isotopic data we collected in 2014 with carbon on the X and nitrogen on the Y. And this method essentially asks, what's the probability of drawing a data point from one species that's within the niche space of another? So in this example, we could ask, what's the probability of drawing a green dot, which is a lake trout, from the niche occupied by those blue dots, which are Chinook salmon? And I should say every data point here is an individual fish. The method, we can draw ellipses that can help us visualize how niche is defined, and in this particular case here, we can see what we've seen in some of the prior figures, with the green ellipse being lake trout being somewhat more unique than the other species. The brown trout appear to have a rather wide niche, and then the other 3 species, the Chinook salmon, coho, and steelhead, are very similar in terms of their clustering and their overlap. And so we can actually pull out the percent niche overlap estimates based on this methodology. And to just guide you to this table, what we're looking at here is the probability of drawing one of these species in the rows from the niche space of the species listed here in the columns. Now, what do these percentages mean? If we highlight the lake trout and Chinook overlap again, we see that there's a 16% probability of drawing the lake trout from the Chinook niche and a 38% probability of drawing the Chinook from the lake trout niche. Both of these percentages are referring to this black area here, which is the overlap between the green ellipse and the blue ellipse. And what this boils down to is the difference in size between those ellipses. The area of overlap this black area, is only about 16% of the lake trout niche, whereas it's about 38% of the Chinook salmon's niche. So to summarize these results, there's a less than 18% probability of drawing a lake trout from the niche space of Chinook salmon, coho salmon, or steelhead, whereas there's more moderate overlap between lake trout and brown trout and very high overlap between the Chinook, coho, and steelhead. And all these results point to competition for declining prey fish will likely be highest among Chinook salmon, coho salmon, brown trout, and steelhead moving forward. Now, I don't believe I have too much time left, but this last slide here just covers some next steps. We're hoping to combine these results with total consumption estimates to model food web effects on prey, things like bioenergetics and ecopath, ecosim models. These results can then help inform the level of consumption on the prey. We continued our collections last year, and we're collecting again this year for gut contents and fatty acid analysis as part of some collaborations with the Illinois Natural History Survey, University of Wisconsin-Milwaukee, and Purdue. This will help us ground truth the diets and evaluate changes in time during this time period of rapidly changing forage. We're also hoping it'll provide some greater resolution on our prey isotopes. And then last year we also provided some tissues to a collaboration with Notre Dame to assess how bioaccumulation and contaminants may be changing with this changing forage base as well. So